Hello everyone, welcome today to this audio podcast. This audio podcast is part of the Serology and Squil. This audio podcast is part of a Hustle and Serotype. This audio podcast is part of a Defcon premiere, and this audio podcast as part of a Storm Logistics production. In this audio podcast today, I'm looking at looking at in around immunobiology and stratifying receptors, and sort of now continuing on. So in early research works, investigators had postulated targeting CXCR3 markedly decreases the tempo and severity of acute allograft rejection, or AAR, in vivo. This effect is now understood as being associated significantly with decreased intragraft accumulation of activated T cells, producing interferon gamma, and impairment of chemokine dependent recruitment of multiple cell types. The investigators had suggested an important link, one of the important links for CXCR3, and in context of clinical transplantation, and potentially in the management of acute in the management of acute allograft rejection, AAR. So during the several renal diseases, or during across several renal diseases or kidney diseases, the infiltration of monocytes and macrophages and T cells into kidneys is thought to play a central role in the progressive interstitial fibrosis and the progression of AAR, leading into CKD, or chronic kidney disease. During the interplay, a crosstalk between cytokines, vasoactive substances, chemokines, and sort of the respective target cells are elicited. The interaction contributes to the outcome. So, for example, healing, or healing, or progression of the particular or specific renal disorder. So chemokines appear to be integral players in this complex and within the dynamic process. So in early research, additionally, rat laboratory models mimicking transplantation have allowed immunobiologists to understand the functional analysis of chemokines in AAR. In early works by investigators, rat laboratory research had informed that the immunological components, RANTES, a messenger RNA, was upregulated by 6 hours during the initial transplant rejection phase, and then again after 3 to 6 days. That draws this audio podcast to a close. This audio podcast as part of the Serology and Squil. This audio podcast is part of the Hustle and Serotype. This audio podcast is part of Defcon premiere, and this audio podcast is part of a Storm Logistics production.